Good morning, good afternoon, and good evening, everyone, and welcome to this Inside Scientific webinar titled Fine-Tune Nutrient Intake Timing for Cardiac Health. I'm Darius Sulam from the events team here at Scientist.com, and I'll be your host today. This webinar has been sponsored by Columbus Instruments, so a big thanks to them for helping to make this webinar possible. Our speaker today is Dr. Martin Young, Professor of Medicine at the University of Alabama at Birmingham. In just a few moments, Dr. Young will be our guide to the fascinating daily rhythms that are critical for normal cardiovascular function and to the role of nutrient intake timing as it impacts the heart. Just before we get started, a few notes on housekeeping. First, if the panels on your screen are too big or too small, you can zoom in or out in your internet browser to adjust their size. Next, the webinar is being recorded, and the video and its associated resources will be available to watch shortly afterwards. We'd love to hear from you and invite you to share any questions, thoughts, or comments in the Ask a Question panel shaking on your screen. We will be taking your questions in the Q&A session after the presentation. If you would like to learn more about the research or technology presented today, explore the resources panel where we've added a few relevant links and downloads. And finally, if you happen to have a technical issue, refreshing the page will nearly always fix things, but if it doesn't, reach out to us using the Ask a Question box and we will help you to troubleshoot. One more thing before we get started, we'd like to hear straight from the audience with a few polls. So the first question I have for you: spillage is a problem when measuring food intake. Choose the option that is most closely reflects your own. Some food spillage is okay, provided the cage is low stress, like an IVC home cage. Natural environment is key. Or accurate FI is paramount, spillage must be addressed appropriately. Some added stress from a novel feeder could be acceptable so long as it can be overcome with acclimation. Give everyone just a few more moments here to get your answers in for this poll. And then I will jump to one more poll here. Are you satisfied with your current system? Options are yes or no. Give everyone just a few more seconds to get your answers in for this poll. Thank you so much for participating. We really do appreciate hearing from you. And that looks great. And now I would like to welcome our presenter, Martin Young. Thanks so much, Martin, for joining us. The floor is yours whenever you're ready.
All right. Well, thank you, Darius. I really appreciate it. I'm also very grateful to Columbus Instruments for giving me the opportunity today to talk about some of our research that really is trying to intersect between multiple fields, and in particular, how nutrient intake impacts cardiometabolic health and how that intersects with chronobiology. So here's just my title slide with some information about myself. And so let's go ahead and get started. All right, so the purpose of this slide really is just to orientate everybody. Just to remind you all that there are these quite dramatic time of day dependent fluctuations in virtually all aspects of human physiology. Whether you're a neuroscientist and you're interested in cognitive function, or whether you're interested in neurohumoral factors, you're interested in metabolism, and the topic of today's talk in cardiovascular function. And so here's a slide I like to start with, and this is a slide that is showing in healthy individuals how cardiovascular parameters change over the course of the day. So on the left-hand side of the slide, you can see this particular healthy individual, when they go to sleep, both blood pressure and heart rate are dropping, and then upon awakening, you see this steep rise in both of these parameters. On the right-hand side of the slide, you can see an average data set for multiple healthy volunteers. Again, you can see that beautiful dipping during the sleep period. And then when someone wakes up, you see this rise in both heart rate and blood pressure. Interestingly, there's a second peak in the early hours of the evening And that's something I'm going to come back to in a few moments' time. Unfortunately, associated with that steep rise in blood pressure and heart rate in the early hours of the morning, there's also a steep rise in the incidence of sudden cardiac death and myocardial infarctions. In fact, it's been estimated that approximately 2/3 of all heart attacks occur between 6 a.m. and 2 p.m. So that's quite a tight window there. Now, classically, these rhythms in cardiovascular physiology and pathophysiology have been attributed to our behaviors, such as sleep-wake and also feeding-fasting cycles, which in turn are impacting a whole host of parameters that are outside of the cardiovascular system, such as changes in autonomic, sympathetic, and adrenergic stimulation, changes in physical activity that lead to changes in peripheral vascular resistance. In addition, as I kind of mentioned, we have periods of feeding and fasting. We do not graze. We are not cows or sheep, and we have set meal times pretty much. And so associated with those meals, we see quite dramatic fluctuations in various neurohumoral factors, such as nutrients, as well as factors such as insulin. So as I've kind of mentioned a couple of times, these rhythms in these factors that are outside of the cardiovascular system really seem to be dependent primarily on various behaviors. So what two very clever scientists decided to do, and this is Frank Shearer and Steve Shea, was they decided to test the question, are factors that are important in cardiovascular function and cardiovascular rhythms, are these factors simply responsive to our behaviors and or our environment? So what they decided to do was they decided to take healthy volunteers and they housed them in an environment that imposed either a 20-hour day or a 28-hour day, depending on the study that they performed. So similar to somebody flying to another planet, for example, and inhabiting that planet that has a different light-dark cycle. What they found was when people were living 20-hour days or 28-hour days, and they were enforcing this by the timing of food intake, as well as the timing of bed rest versus physical activity, as well as, of course, the light-dark cycle, they found, to their surprise, that they still observed, in many cardiovascular parameters, 24-hour rhythms. So even though behaviors and the environment had either a 20 or a 28-hour cycle. These are things such as that early rise in cortisol that we see in patients. That was still occurring over a 24-hour period, even though these individuals are living 20 to 28-hour days. Heart rate was still increasing in the equivalent of the early hours of the morning. And if you recall, I mentioned that there's a second peak in blood pressure in the early hours of the evening in healthy volunteers. Well, that secondary peak seems to be driven by endogenous clocks. And the first peak that you see usually when you wake up, that seems to be driven by behavior. That rise in blood pressure in the early hours of the morning is probably secondary to the fact that you're waking up. But clearly, there are 24-hour rhythms that are driven by some endogenous system that is independent of our behaviors and our environment. We now know that endogenous system is the so-called circadian clock. Many of you, when you think about a circadian clock, you might think of something that is found in the brain that is regulating, for example, sleep. And you'd be absolutely correct. There is a master clock within the brain that regulates sleep. But it turns out that these clocks are found everywhere in the body. In fact, every cell that has been investigated in mammals, pretty much, a clock has been found. So what is this clock? Well, clocks are intrinsic to the individual cells. They are a transcriptionally based mechanism that are made up of positive and negative feedback loops. So right here in the center of this slide, you'll notice that there are two proteins called clock and BMAL1. These are transcription factors. And when they bind together, they can activate the transcription of genes by binding to E-boxes and promoters. This includes components of negative feedback loops the period and the cryptochrome genes located on the left-hand side of the slide, as well as reverb alpha and reverb beta on the right-hand side of the slide. When these proteins accumulate, they will inhibit the clock BML1 heterodimer and therefore act in a feedback loop manner. As I've mentioned already, all of these factors are able to modulate transcription. So in addition to regulating themselves, they also are regulating a whole host of other genes within the cell that in turn is going to change cellular function in a time of day dependent manner. So what we decided to ask many years ago, I believe it was in the early 2000s, we decided to ask the question, is there an endogenous clock within the heart cells that might be affecting heart function? And those heart cells, of course, are cardiomyocytes. So in order to address this, we took heart cells from rodents, we placed them into a culture dish, and we monitored the activity status of these various clock components. And as you can see here on the right-hand side of the slide, even though these cells are in isolation in a culture dish, you can still see 24-hour rhythms in their expression or their levels. And that the timing of these oscillations are identical both in cell culture and in vivo in an intact heart seen on the left, where BML1 is peaking about 12 hours before DBP, which peaks just before PER2. So at this stage, we were happy in saying, okay, there is a clock located within cardiomyocytes. What is it doing? Well, in order to answer that question, we do what many scientists do, and we say, you know what, let's get rid of it. Let's remove it from the system, and let's see what the consequence is. And that might give us some insight with regards to what it usually does. And so that's exactly what we did. We targeted this central clock BML1 heterodimer, which was at the heart of the clock mechanism. In the first model, termed CCM, what we did was we overexpressed only in cardiomyocytes a dominant negative clock mutant protein that is able to bind to BML1, and that heterodimer can bind to E-boxes and target genes, but cannot induce transcription, and therefore the clock mechanism falls apart. In the second model on the right-hand side of the slide, you can see that we generated a cardiomyocyte-specific BML1 knockout using a classic Cree-Lock system. So here we essentially, as it implies, we knocked out BML1 only in cardiomyocytes, and this model is called CBK. We then performed microarray studies on hearts that were isolated from wild type and CCM or wild type and CBK hearts at different times of the day. And as you can see from these slides, from this particular slide, that in wild type hearts, we see these beautiful time of day dependent oscillations in gene expression that are completely lost in both CCM and also CBK hearts. And using this strategy, we've estimated that anywhere between 5 to 10% of all genes that are expressed in the heart are somehow regulated by the cardiomyocyte clock. When we go on and we perform gene ontology analysis on these genes, we find that the clock seems to be regulating processes such as electrophysiology, cellular signaling, and cardiac metabolism. And as we know, all of those processes in turn regulate contractility of the heart. Well, is there any evidence that the clock regulates contractility of the heart? Indeed, there is. So we interrogated this in vivo through the use of telemetry, in vitro using isolated working mouse hearts, and in collaboration with Tammy Martino in Canada, even in vitro. So first of all, on the left-hand side of the slide, the telemetry studies show that for the mouse, which is nocturnal and therefore has increased activity during the dark period, you see these beautiful rhythms in heart rate with a peak heart rate in the middle of the dark period or the active period, and that these oscillations are attenuated in our clock mutant hearts, and they exhibit bradycardia or low heart rate. In the middle panel, you can see that when you take hearts out of the animal and you looked at contractility of the heart, here evidenced by cardiac power, even though there's no neurohumeral stimulation ex vivo, you can still see in the control hearts these very nice time of day dependent oscillations with a peak contractility in the middle of the active period. And in the clock mutant hearts, that's flatline. There's absolutely no time of day dependence. And then in vitro, when you look at myosin ATPase activity, the same thing is seen. Peak activity during the dark period, the active period in control hearts, and in the clock mutant hearts, this is flatlined. So at this stage, we're happy in saying that the clock in the heart is regulating a whole host of genes, and that the clock in the heart is regulating cardiac contractility. My lab is particularly interested in cardiac metabolism, and we know, as you can see here illustrated on this slide, that it's a long way from changes in gene expression to changes in cardiac function through metabolism. You have to have an alteration in the protein, changes in the enzymatic activities, and ultimately changes in metabolic flux. So is there any evidence that these steps in between are clock regulated? The answer is yes. So again, Tammy Martino in Canada, what she did was she took hearts from our CCM model and she performed a proteomic study. And these were hearts collected at different times of the day from control versus our clock mutant hearts. And as you can see here, that there was a time of day dependence in about 4% of the cardiac transcriptome, the cardiac proteome, and that this was impaired when the cardiomyocyte clock was ablated, so in the CCM animals. And interestingly, many of these proteins are involved in cardiac metabolism. So we again collaborated with a colleague in Canada, this time Christine Desrosiers in Montreal, who performed metabolomics for us. And as you can see here, both lactate and malate, as well as many other metabolites, have beautiful time of day dependent oscillations in control hearts. And in our CBK model, our BML1 knockouts, it's flatline. We lose these time of day dependent oscillations. Here's an example I really like. This is ketone body metabolism, focusing in on one of the key enzymes in ketone body metabolism, that being BDH or BDH1. As you can see here in both our models, the CBK and the CCM model, there is only about 10% of the mRNA remaining in the hearts, in the clock mutant and the BML1 knockout hearts. And then when we look at protein, we see a dramatic attenuation. In activity of BDH1, it's almost gone. And when we look at ketone body oxidation flux, there's a significant attenuation. It's down to only about 1/3 that's seen in a control heart. All right, so what I'm going to do here in this slide, I'm going to attempt to show you about 15 years worth of data in one slide. All right, and this is data in which, through the use of various strategies, including radio-labeled tracers, we have studied metabolism in the heart at different times of the day. And we've also looked in our clock mutant and our BML1 knockouts to see, is the cardiomyocyte clock driving these rhythms? So all the data here is clock-mediated cardiac metabolism oscillations. What we find is at the beginning of the active period, this is a very special time. This is the time of increased oxidative metabolism in the heart. And here, particularly, you can see that there's an increase in the oxidation of glucose at the beginning of the active period, and to a lesser extent, fatty acids too. Now, this might make sense. Okay, so when the animal in the wild wakes up, there's going to be an increased energetic demand on the heart because it's got to increase heart rate, and it's got to increase contractility. And so that increased energetic demand seems to be met by an increase in oxidative metabolism. When you go now towards the end of the active period, this seems to be a time at which excess nutrients that have been taken in during the active period are channeled into storage forms such as glycogen and triglyceride that we believe is in anticipation of the upcoming sleep period when the animal will be fasting. So the heart has enough nutrients to maintain contractility in this basal state. And then at the beginning of the sleep period, this is also very interesting. The beginning of the sleep period, this is a time of growth and repair of the myocardium. So where you have high rates of cellular constituent turnover. You get rid of damaged proteins, phospholipids, and organelles, and you replace them with new in anticipation of the upcoming awakening of the organism. So at this stage, you might be thinking, okay, well, that's very interesting. All right, so he's shown all of this, but I've got a lot of questions, and we have a lot of questions too. And two of those questions that to us were kind of obvious was, first of all, what are some of the mechanistic links between the clock and these changes in cardiac parameters such as contractility and metabolism? Well, we are studying this, and here on this slide I'm showing that one candidate we're studying right now is E4BP4. I'm not going to talk about that today. Instead, I want to talk about the second question, and that is, what are some of the functional implications to all of this? So you might be wondering, is there any situation in which the clock becomes dysfunctional and that perhaps leads to disease or contributes towards disease? And that's what I'm going to show here. So we know that the clock is regulated by almost every environmental factor you can think of and almost every behavior, ranging from changes in light exposure to diet, and this is quantity, quality, as well as timing, sleep duration and quality, physical activity, aging, as well as various factors such as cortisol and sympathetic tone. And many of these things change in disease states. And there's a lot of evidence now from multiple groups, including our own, that if the clock becomes dysfunctional, it leads to disease. And the best example in humans is shift work. Shift workers, without question, they have disruption in their circadian environment and in their behaviors. And unfortunately, they have increased risk of various pathologies, ranging from cancer to obesity, diabetes, mental or cognitive disorders, as well as cardiovascular disease. And here on the right-hand slide, I'm just showing you an example of a meta-analysis indicating that shift workers have about a 25% increased risk of having heart attacks compared to day shift workers. Well, what about the cardiomyocyte clock? So when we disrupt the cardiomyocyte clock, and in this case I'm showing the BMAL1 animals, so the CBK animals, you can see here that with age, we get a hypertrophic phenotype as depicted by an increase in cardiomyocyte size. These animals develop a pronounced cardiac fibrosis, as well as with age, cardiac dysfunction occurs and they develop heart failure by 6 months of age. And in fact, by 1 year of age, all the animals have died, and they have died due to dilated cardiomyopathy. So that's a very, very severe phenotype. And clearly, therefore, the clock is essential for maintenance of normal cardiac function. All right, so here's just a little summary of the first half of the talk before I move to the second half of the talk. Hopefully, I've convinced you that there is a clock within the heart and that it does some good, that it is important for temporally partitioning various processes, including cardiac metabolism, and it's important for maintenance of cardiac function because when it's disrupted, it leads to decrease in contractility of the heart, adverse remodeling, and ultimately heart failure and even death. So that's the good and the bad. Here's something that we've started to ask, and that is there ever a situation in which these circadian clocks can contribute towards disease? Perhaps in response to the way in which we live our lives or our environment? And the answer seems to be yes. So let's go back to this slide right here that you've seen multiple times. This is the clock control of metabolism in the heart. And it turns out that this is also true for other tissues as well to a large extent, although the timing might not be exactly the same in different tissues. We have begun to ask the question, okay, well, if the heart is performing metabolism in a time of day specific manner, if we challenge the myocardium or even the whole body with specific nutrients at different times of the day, will that either lead to an adaptation or could it lead to a pathology? And what we did at first was we focused in on dietary lipid or high-fat diets, as many people in the field do. So here's just a little bit more information before I start to show you some of those particular experiments. This is what we knew at that particular moment in time. And some of this information I've already shown you. So this is what I'm doing here is I'm focusing in a little bit more detail about how tissues, including the heart, respond to fatty acids in a time of day specific manner. I already told you that at the end of the active period, that's a time of increased excess nutrient storage. In the case of fatty acids, when they are in excess, they are stored as triglyceride. Well, unfortunately, if that occurs over and over and over again, that can lead to an excess amount of lipid within the cell known as steatosis. And of course, if that happens in fat cells, that increases your adiposity and leads to weight gain. If it happens in the heart, it can lead to contractile dysfunction of the heart. And if it happens in other organs, such as the liver and in skeletal muscle, it can also lead to dysfunction of those tissues. I also mentioned that in the sleep phase, this is a time of cellular constituent turnover. And so that includes an increased synthesis of phospholipids. Unfortunately, that means if you then provide an excess amount of fatty acids at that time of the day, you can get an imbalance in lipid signaling species, such as an excess amount of phospholipids and cholesterol ester, which is associated with lipotoxicity and is detrimental. Conversely, and what I haven't told you so far is the beginning of the active period. This is a time at which, if you are challenged with fatty acids, you can switch on the genes involved in the oxidation of those fatty acids, and you can actually increase the capacity to burn fat as a fuel, which is good. It would be great if we could burn more fat as a fuel because it would be beneficial for diabetes, obesity, as well as cardiovascular disease in general. So based on this, we hypothesized that if we force, and these of course are rodent studies that we were doing this in, if we force the rodent to consume lipid-rich meals at the end of the active period or during the sleep phase, that could be detrimental in terms of both cardiometabolic disease as well as cardiac dysfunction. How do we test this? Well, these are studies that go back to around about 2008 and 2009 that we performed in collaboration with Molly Bray, who is currently at UT Austin. What we did was we performed a large number of different feeding studies that you can see on the left-hand side of the slide, in which we fed animals either a control diet, low in fat, or a high-fat diet at specific times of the day. We did this through the use of our comprehensive laboratory animal monitoring system from Columbus Instruments that we had specialized, custom made. And our system is a wonderful system. It was essential for these studies because we had a system generated in which we have 2 feeders in each mouse cage so that we could have one feeder that houses a low-fat diet and another feeder that has a high-fat diet. In addition, our system has computer-controlled food access capability, meaning we can program a computer to open and close feeders in a time of day dependent manner. So the mouse can only access either the low or the high fat diet at specific times of the day. In addition, we're able to monitor the food intake through the use of highly sensitive balances on both those feeders, so we can see how much of the diet the animals are consuming. We also have a beam break system that allows us to monitor physical activity, as well as performing indirect calorimetry that allows us to monitor energy expenditure, or as today I'm going to show you, allows us to monitor the respiratory exchange ratio, which is an indicator of which substrates the animals are burning as a fuel. Okay, so what I'm going to do is I'm going to focus in on the bottom left-hand corner of this slide. You can see this feeding regime at the very bottom. This is a feeding regime in which we fed the animals either a high-fat meal at the beginning of the active period or a high-fat meal at the end of the active period. So basically a high-fat breakfast or a high-fat dinner. And we wanted to know what impact did that have on, first of all, cardiometabolic parameters. Here's the data. So what we found, first of all, was, and here you can see that there was absolutely no difference in the total amount of food that the animals consumed or the total amount of fat that the animals consumed. The only difference between these two groups, in black, the early high-fat meal, and in red, the late high-fat meal, the only difference is the time of day at which they are consuming the lipid-rich meal. When we then look to see, using indirect calorimetry in our clam system, we look to see which fuels the animals are burning at the whole body level, we got this staggering result that to this day still blows my mind. What we found, first of all, for the group in black, and this is the animals eating the high-fat meal at the beginning of the active period, we saw that the animals had metabolic flexibility, and we got the result we absolutely anticipated. So I'm going to take you through this. Immediately at the timing of the consumption of that high-fat meal, and that is at time 12 that you can see on the x-axis, you can see the RAR increases to an intermediate level. And that's an indication that the animals are burning both carbohydrates and fat as a fuel. At the end of that meal, the animals start fasting, and so the RAR drops because they're now burning fat. And then at the end of the active period, when they have the high carbohydrate meal, you can see the RAR increasing. That's because they're burning carbohydrate as a fuel. So that's perfect metabolic flexibility. The animals are utilizing the substrates that are being provided in the diet. Here was the thing that really was very surprising. For the animals that started the active period by eating a low-fat meal, high carbohydrate meal, you can see the RQ goes all the way to a value of 1, consistent with burning carbohydrate as a fuel. However, during the 4-hour fasting period in between, the RAR did not drop. And then when they had the high-fat meal, the RAR also did not drop at the end of the active period. They developed metabolic inflexibility. So the animals that are consuming the high-fat meal at the end of the active period are unable to respond to that acutely. Was there any consequence to this? Well, the animals that are eating the high-fat meal at the end of the active period gained more weight, they had an increase in adiposity, and they developed glucose intolerance. They also developed hyperinsulinemia as well as dyslipidemia. Essentially, they developed cardiometabolic disease, even though they were eating the same amount of fat and the same number of calories compared to the animals eating the high fat at the beginning of the active period. So we went on several years later and we decided, well, to look at the heart. So that was whole body parameters and cardiometabolic health. What about specifically the heart? And so we repeated the studies, again using our clams. And you can see here when we repeated the studies, once again, there was no difference in the total amount of calories consumed or the total amount of fat consumed. The only difference was whether they were eating the fat early or late in the active period. When we looked at contractility of the heart, we found that the animals that were consuming the high-fat diet at the end of the active period, they had a decrease in cardiac contractility. They were beginning to develop cardiac dysfunction. And this is after only 9 weeks of performing this intervention. More recently, what we've done is we've tried our best to encourage animals to eat a high-fat meal during the sleep period or the inactive period. This is the light phase for the rodent. By providing that high-fat meal only during the light phase. And as for a contrast, we had another group where we fed the animals a high-fat meal during the active period. When again you use the clams to enforce this, and you look at the consumption of these diets, you find that the animals that are provided the high-fat meal during the sleep period, what they do is they essentially chow down at the very beginning of the sleep period. They have a quick meal, and then they go to sleep. And then they do not consume any more of the high-fat meal. Maybe a little bit at the end of the sleep period, but that's about it. Be equivalent to having a very cheesy pizza before going to sleep at midnight. In contrast, the animals that were given the high-fat meal during the active period in red, you can see that they consume a large amount of dietary fat. And so you might think, well, okay, well then they're probably not doing very well when you look at their hearts, because fat is supposed to be bad for the heart. Not the case at all. So the animals in red that were provided the high-fat meal during the active period, their cardiac contractility was better compared to the animals that just had that high lipid meal at the beginning of the sleep period. And that was associated with a better ability of the heart to oxidize the fat as a fuel. Okay, so far what I've done is I have shown you data in which we have tried to prevent disease. Okay, in which we have done interventions right at the start to see if animals behave in one way versus another. Can we prevent cardiac dysfunction, or can we precipitate cardiac dysfunction? But that doesn't always tend to be the way things happen in the clinic with humans. Instead, what tends to happen is people, when they're young, tend to eat however they wish, because they can. And then later in life, people develop adiposity and diabetes. And then they start to ask questions, what can I do to prevent myself from developing heart disease? And so that is a treatment strategy. And so in this recent study, what we did was we got animals nice and fat by feeding them high-fat diets. They could eat it whenever they wanted. You can see in the red group, this is ad libitum, high-fat feeding. They develop adiposity. They also develop markers of adverse cardiac remodeling. The heart gets bigger, there's more fibrosis, and there's more lipid within the heart. This is the beginnings of cardiac obesity cardiomyopathy, or cardiac remodeling. We then ask the question, okay, when an animal is at this stage, can you reverse this? Can you now do an intervention where you can prevent the development of dysfunction? So what we did was, for the last 2 weeks of this study, we kept the animals on the same diet, but we prevented them from being able to eat the high-fat diet only during the sleep period. Because remember, the sleep period seems to be a period where you should not be eating a high-fat meal. And when we do this for just 2 weeks, we see we completely reverse the steatosis, we reverse the cardiac hypertrophy and enlargement of the heart, and we attenuate the fibrosis. However, the animals are still obese. So we have not reversed the adiposity, but we have reversed the beginnings of the cardiomyopathy. Again, we did these studies utilizing our CLAMS system, and when we looked at the RER data, we found something that was very interesting to us. We found that in obese animals, they develop a severe metabolic inflexibility over the course of the day. So if you look at the panel on the top here, these are animals that have been fed either the low-fat or the high-fat diets in just a free fashion, so ad libitum. The control animals, they're lean, and you can see these beautiful 24-hour rhythms in metabolism over the course of the day. And in the obese animals, it's almost as if they're dead. It's flatlined. There's absolutely no rhythms over the course of the day in terms of metabolism. When we then do this intervention for 2 weeks of time of day restricted feeding, we breathe life back into metabolic flexibility. You can see both in the controls, look at the size of those metabolic oscillations, and even in the obese animals, we start to see metabolic oscillations once again. When we look at the hearts, we see this is true there as well. During obesity, we lose our metabolic flexibility in lipid metabolism, and we can bring that back when we perform time-of-day restricted feeding. And that is seen when you look at both cardiac triglyceride synthesis as well as when you perform a lipidomics approach. Okay, so all of this together, we're happy in saying that fatty acids are consumed at the beginning of the active period. It seems that the myocardium can adapt and it doesn't lead to contractile dysfunction. However, when fatty acids are consumed at the end of the active period and or the sleep period, it leads to adverse remodeling and cardiac dysfunction. In addition, it also leads to cardiometabolic disease. Now, when we first published these data, we were ridiculed a little bit by the British press. They love to ridicule people a little bit. And we got a large number of headlines, as you can see here. And we even, in one newspaper in England, when we published these studies, we got a little cartoon here in which the individual is saying, I'm trying to lose weight, so I'll go for the full English breakfast. There you go. Well, fortunately, it doesn't quite work that way because a full English breakfast is also an increase in caloric intake. Our whole idea here is that it's, if you keep the calories constant, when should you eat the lipid? And we're suggesting that it should be at the beginning of the active period, basically for breakfast. Now, luckily, since our study, a large number of individuals and labs around the world have replicated these findings, both in rodent models as well as in humans as well. So much so that the American Heart Association released a scientific statement back in 2017 that recommended eating a greater share of total caloric intake earlier in the day to have positive effects on risk factors for heart disease and diabetes mellitus, essentially cardiometabolic disease. Now, interestingly, they did not say anything about lipid. They said total caloric intake. In our studies, of course, we've always been focused on lipid. Got us thinking, should we also be thinking about other macronutrients? And so recently we've turned our attention to protein, because everywhere you go, you find that people are saying, I'm going to reduce my carbohydrate intake and my fat intake, and instead it's better to have protein. So we decided to ask the question, in terms of cardiometabolic parameters, is it better to consume protein at the beginning of the active period or at the beginning of the sleep period? Is there any kind of benefit to that? Now, we actually at first focused in on a specific type of amino acid that is enriched in red meat, and those are the branched chain amino acids. And we focused on the branched chain amino acids because an elevation in branched chain amino acids is associated with disease in humans. It's associated with obesity, diabetes, and also cardiovascular disease. In addition, we are able to see a very easy readout after we challenge the heart with branched chain amino acids. And that is essentially the activation of this mTORC pathway and an increase in protein synthesis and growth of the myocardium. So again, using our clams, we decided to ask a very crazy question. I didn't think this was going to work. We asked a crazy question. We said, if we feed rodents just one single 4-hour meal that is enriched in branched chain amino acids, will that lead to a growth of the heart? Is that enough time to change the heart? Well, first of all, we said, let's feed them this BCAA enriched meal at the beginning of the active period, so for breakfast. And when we do that and we look at the heart cells in the animals, we find that there's no growth of the heart whatsoever. Okay, I didn't think that we would. It was only 4 hours, right? We repeated the study, and we did this now at the end of the active period, so a high BCAA meal for dinner. When we do that, we see within 4 hours there is a 70% increase in cardiomyocyte size. This was stunning. And we had to do this so many times. I didn't believe it at first. We ended up doing this 30 times and every time we saw the same result. Well, you might be wondering, okay, that could just be a swelling of the cells. So maybe they're taking in more water. Well, we looked at the wet weight to dry weight ratio, and that was not the case. There was no difference. We think it's protein synthesis because then when we look at protein synthesis, we find that branched chain amino acids can only activate protein synthesis in the heart when consumed at the end of the active period. No effect whatsoever when they're consumed at the beginning of the active period. And if you inhibit protein synthesis, you block the growth of the heart. So this is due to protein synthesis. So we then said, okay, well, if after one meal the heart's going to grow 70%, what if we did this every day for 4 weeks? Can we see an accumulative effect so the heart just keeps on growing and growing and growing until eventually it might lead to dysfunction? And so again, using our clams, we did this. We did not see that there was an accumulative effect. Instead, at the end of those 4 weeks, when we looked at hearts from animals that were fed the branched chain amino acids at the end of the active period every day for 4 weeks, heart size or cardiomyocyte size was 70% increased again. So it suggests to us that it's transient, that the cardiomyocytes get bigger in response to the meal, and then over the course of the day, they shrink back down. And then they're going to get bigger again, and then they're going to shrink back down. And this was associated with an immediate activation of this mTOR pathway, only when branched chain amino acids are consumed at the end of the active period. Okay, well, that was in a healthy animal. So that might be for healthy people on this call. This is fine. And in fact, maybe there's some benefit to this. But we then asked the question, well, what if we look in an animal model of disease and heart disease? Does that lead to detriment, this kind of lifestyle? And the answer was yes. So what we found was when the animals consume every day for a 6-week period, this high branched chain amino acid diet at the end of the active period, and the animal has heart disease, you see an acceleration of heart disease development, as shown here on the far right-hand side, very bottom. Ejection fraction is dropping. These animals have heart failure. And so we published this study at the end of last year in which we're basically finding that the time of day at which branched chain amino acids were consumed dramatically affects the growth of the myocardium, and it could be detrimental during heart disease. Is there any evidence in humans? Well, in terms of heart disease, no, not yet. But in terms of cardiometabolic health, yes. So this is a study published by the N. Haynes group that is showing that individuals who consume protein-rich diets for breakfast, they have better cardiometabolic health than people who are consuming proteins for dinner. All right, so here's my summary slide. So hopefully I've convinced you that within every cell in the body, there is a clock mechanism, which includes the cardiomyocytes of the heart, that are driving rhythms in various physiologic parameters, such as metabolism, and that can interact with our behaviors, such that if you were to consume meals at the wrong time of the day, that can interact with these clocks and could lead to dysfunction. In addition, I didn't have really much time to talk about these other parameters, but I did touch at the beginning on this concept that there is this time of day dependent rhythm in the onset of adverse cardiovascular events such as heart attacks and sudden cardiac death, as well as there are other implications such as diagnostic accuracy, outcomes, therapeutic interventions such as what's the best time of the day you should take medications. This is known as chronopharmacology, to avoid side effects. What's the best time of the day to perform your studies? That's maybe during the discussion period. And then can we target these clocks for the treatment of cardiometabolic diseases? And right now, the answer seems to be yes in preclinical models, that when you target these clocks, for example, with reverb alpha agonists, it seems to be beneficial. Okay. With that, I'd just like to thank all the individuals that aided with these studies, including multiple collaborators here at UAB, as well as individuals in the lab, and collaborators elsewhere, including Mitch Lazar, Christine DeRosier, Tammy Martino, Abhinav Dewan, Sumanth Prabhu, and Molly Bray at UT Austin. And with that, I'd like to thank you all for your attention. And of course, once again, I'd like to thank Columbus Instruments for all their support over the years, who have really been instrumental in making these studies possible. Thank you.
Hi, Martin.
Hey, Darius. How you doing?
Great. Thank you very much for your presentation once again.
Oh, thank you.
All right. So let's get started here. First question I have for you. For the time of day feeding studies, do you think the cardiac benefits observed when nutrients are restricted towards the beginning of the active period are due to direct effects on the heart or secondary to systemic effects?
That's a great question, Darius. Thank you very much. To be honest, I think that it's a combination of the two. So whenever you perform these whole body studies, so these in vivo model studies, you are going to have alterations in a large number of organs. And so, as I already showed in some of the studies, that when you perform this time of day restricted feeding, that you are impacting a whole host of cardiometabolic parameters that in turn are going to have influences on the myocardium. So by doing this time of day restricted feeding, one of the things that we think is happening is that you are helping to synchronize the heart with neurohumoral factors, as well as all the other organs in the body. Because during obesity, it seems that you develop an internal dysynchrony or a circadian misalignment seems to be happening, where different organs, unfortunately, aren't necessarily doing what they're supposed to in terms of temporal control. And so you get alterations in normal rhythms in various parameters. So I think it's a combination.
Great. Thank you so much. Here's another one for you. What are your thoughts on the effect of types of fatty acids, saturated and unsaturated, and their effect on metabolic flexibility / cardiac health?
That's a very good question. So clearly, there are quite a large number of fatty acids, and saturated versus unsaturated are two classifications. Classically, when we do these animal-based studies, we're often giving a large proportion of saturated fats. These saturated fats are probably more detrimental when we are looking at parameters such as cardiometabolic dysfunction and also cardiac dysfunction. And that's going to be through a large number of mechanisms. And so, of course, some are things like very direct, things like palmitate, which is a saturated fatty acid, can go on to make signaling molecules like ceramides. And those in turn lead to toxicity, lipotoxicity in cells, and sphingolipids as well. So to answer your question, there's definitely the evidence suggests that saturated fats are more detrimental. But it's such a heterogeneous population there that there's a lot of things going on.
Wonderful. Intermittent fasting is a hot topic. Have you looked at intermittent fasting on your outcomes? And if not, can you speculate on this?
Yeah, it's a really good question. So, intermittent fasting is a very heterogeneous term. Different people perform intermittent fasting in different ways. Some folks will fast for an entire day, and then the next day they'll eat, and then they'll fast for an entire day, or so on and so forth. Some people perform intermittent fasting where they only allow themselves to eat in an 8-hour window during the day, and then for the remaining 16 hours, they're going to fast. And without question, intermittent fasting has benefits for cardiometabolic disease, and even now it seems cardiovascular outcomes as well. One thing we would like to understand is what is the connection here with these circadian clocks, and does the time at which you break that fast, so basically the period during you're consuming food, say for somebody who's eating 8 hours a day and fasting for 16 hours, is it important to consider what time of day that you are actually consuming the diet? It's a little bit more tricky to do in a rodent, I'm not going to lie. I don't know if you noticed, but for many of our studies, we tried not to fast the rodents, at least more recently, for longer than 4-hour periods at a time. And that's simply because of the very high metabolic rate of rodents, that it's difficult to equate what an 8-hour, well, actually a 16-hour fast in the human is not the same as a 16-hour fast in a rodent. Not at all. I think the rodent's metabolic rate is about 8 times faster than humans. And so we have not been trying to model that fasting for an entire day and then feeding and fasting and feeding. But there are benefits.
Yeah, for sure. Thanks so much for that one. Here's another one for you. We're running out of time here, but I'll try and squeeze in as many as I can. While you did not show the effects of carbohydrates, we have shown that even low glycemic index foods taken at dinner have a worse postprandial glycemic response than a high GI meal taken during breakfast. So could you please comment on whether early time-restricted feeding in general than later time-restricted feeding?
Yeah, that's a wonderful question. Certainly, the time of day at which nutrients are consumed is going to then be influenced by digestion and absorption. So in the case of carbohydrates, if you consume them for breakfast or for dinner, you're going to have a very different pattern in the spike of glucose in the circulation after that meal. And also then that's going to involve also then the response of the beta cells to help bring the glucose back down. So the patterns are going to be very different. So for our studies, we focused mainly on how much the animals were consuming, and we did not look at digestion and absorption. And this is something that is absolutely circadian regulated, both carbohydrates as well as fatty acids as well. Digestion and absorption is undoubtedly affected by the time of day. And so you bring up a wonderful question there. Thank you.
I think we have time for just one last more question here. So how does the data translate clinically? Should lipids and proteins be consumed all as the very first meal? And what should we consume at dinner as best for cardiac health?
Well, so again, there haven't been a large number of highly controlled clinical studies. So right now I can only show you the data we have for rodents, as well as then the few studies that have been done in humans. And usually the studies in humans were done in healthy populations. Some studies have been done in obese individuals to look to see whether there's any weight loss. But I am not aware of any controlled studies that have been done in patients that, for example, have heart disease. There are epidemiologic studies, and that I really didn't talk about today, that have tried to understand in individuals that, say, skip breakfast, what is their risk of cardiovascular disease. And again, once again, risk of cardiovascular disease is higher in individuals who skip breakfast. But those aren't causative studies. unfortunately. They're epidemiologic observations. So we really need to do more to understand this. We need highly controlled clinical studies that are translating some of the work that we, as well as others, are doing to see how specific patient populations would respond to time of day feeding. And really, we just don't know at this stage. So I'm very cautious about of how to answer this question in specific patient populations. So
For sure.
More needs to be done.
Definitely a lot of variables involved. I think we're going to wrap it up here. I thank you again so much for this very interesting presentation, Martin, once again.
Thank you very much for the opportunity. I really enjoyed it. Thank you.
And with that, I'm going to bring this webinar to a close. A big thanks to the audience here for participating, and a big thank you, of course, to our sponsor, Columbus Instruments. In closing, we hope you enjoyed this Inside Scientific webinar, and we'll see you again next time. Have a great day, everyone.